Good afternoon, everyone, and good morning and good evening for some people in the Zoom. My name is Meihua Gong. I'm a PhD student of University of Technology Compiègne. It's my great honor to be here to present my project. Today, I will share my project to develop a new tool to purify methylated peptide from bacteria to study bacterial mechanism and some results with you. So firstly, I will present the background of this project. In the environment, for most bacteria with especially some bacteria with flagella adhesion is the first step in its colonization, invasion, and biofilm formation. And bacterial adhesion is a mobility transition from planktonic stage bacteria to sessile stage bacteria. As we can see, the process of bacterial colonization is a process from free swimming stage bacteria when it adheres to the surface of some biotic or some unbiotic surface, and it begins to secrete extracellular polymerase substance, and subsequently to develop biofilm and eventually to form mature biofilm. And for bacterial mechanism, it's also a changement of different stage bacteria, for free floating bacteria to sessile stage bacteria. So for my project, the goal is to identify the changements of different states of bacteria. Here we will focus only on semilative murine. And as for the difference of different states of bacteria, there are some hypotheses trying to explain it. The typical one is that maybe is involved with some post-translational modifications of proteins happen to bacterial flagella. And for our research target, Salmonella is a good example for us to study bacterial flagella. As we can see, the structure of the membrane of Salmonella cells is full of flagella. And the flagellum is a unit of flagella. It's composed with a filament cap, of a long helical filament, and several both body rings, and a hook filament junction, a hook, a type 3 protein export, and a stator unit. And for bacteria in the environment, it's most of them, they change the orientation is by running and tumbling to change the swimming direction. And this process is mainly involved with the filament. So filament could be a good research point for us to study flagella and for Salmonella mechanism. As we mentioned before, the different states of different states bacteria is mainly associated with some post-translational modifications, and the typical one is methylation. And recent years, some research found that methylizing can promote bacteria adhere to the surface or receptor while hydrophobicity. As we can see, the structures of methylizing is mono, D, and tri-methylizing, the three forms of methylizing. And so they could work as a methylated target for us to study methylizing and for us to identify how methylizing are impacted by flagella mechanism in wild type Salmonella and its mutants. To get this purpose, we have to get some purified peptide, methylated peptide. So we have to find a tool with a high affinity, with a high affinity and specificity to purify peptides containing methylizing. Here we are trying to use DNA aptamers. To select specific aptamers, the traditional methods is selects. And since selects technique was established in 1990s, there are numerous variants of selects methods. But they are not all appropriate for our small target molecular methylating. As we can see, the first group, they are not suitable for small target molecular. And the second one, even they can used for small molecular, but when they are used with a high cost, since the small molecular have to immobilize on gold electrode surface. And the third group, even they are easy handling. Even they are easy handling, but when the target in a very small amount, but they shared a common disadvantage that they are not real-time monitoring during aptamer enrichment. And no, okay. And the last group, they only used for cell size, but not tested for small target molecular yet. So for our case, the select technique is combined with fluorescence activated selects and magnetic bead selects. Is called Flumax selects. And during Flumax selects, the target was immobilized on magnetic beads. And meanwhile, with using fluorescence labeled oligonucleotides. So in the Flumax selects, we firstly immobilize different types of methylizing on the magnetic beads to form activated beads. And for the combinational library, when it folds into functional oligonucleotides, and we immobilize with the activated beads, and then we wash away the unbound oligonucleotides. And for the okay, cool. As for the ssDNA, which coated with the beads, we did elution by heat treatment to get the eluted ssDNA, and then by amplify by PCR to get PCR products, and the PCR products will be separated by phage gel to separate to get new ssDNA pool for next rounds. As for the eluted ssDNA from last round, we will be treated with cloning and sequencing analysis. And during this process, we did several rounds positive selection and a round negative selection with negative beads. As for the positive selection, the selected ssDNA, which is coated with beads, but for negative selection is what we collect is the unbound oligonucleotides. And after several rounds positive selection and negative selection, we got 3 rounds selected ssDNA pool against the mono, D, and trimethylation. As we can see, the first 2 graphs illustrate the selection to mono and demethylation. We did 5 rounds positive selection and 8 round negative selection with negative beads at the last round. During the positive selection, we increase the selection pressure at 5th rounds by adding other kind of methylizing and freelizing in the incubation buffer. As we can see, the elution yield of the selected ssDNA during the positive selection is an increasing tendency, which illustrates enrichment of Aptamers. As for trimethylizing, we only did 3 rounds positive selection and a round negative selection with negative beads. And for the selection results, we can see the elution yield of selected ssDNA also in an increasing trend, which also demonstrates a good enrichment of aptamers. And for all the selected ssDNA, we will focus only on the last round. And for all the selected ssDNA from last round will be cloned into plasmid. And finally, we just pick up 20 clonies for further characterization analysis. So firstly, we get sequencing analysis of all the selected ssDNA. As we can see, we get most interesting results is for trimethylizing. As we can see, for all 20 clones, we got around the redundancy around 30%. And by alignment analysis, we can say there's 6 clones share the similar sequences, and we will focus on with high redundancy sequences and including flanking sequences that we named Aptamers. As we can see, the sequence here in the middle region in red color is high redundancy sequences, which is selected by our select process, and at end flanking sequences is which was used for aptamer, which was used for PCR amplification. And we also get good results for mono and demethylizing, even though they are not with high redundancy as trimethylizing, but they are also good. We got around 10% redundancy for monomethylizing, and for demethylizing, we got around 11% of redundancy. And finally, we also synthesized the high redundancy sequences, including the flanking sequences named Aptamono 11 and Apta-D4. As we can see, the sequences here showed in the same color code is with a high redundancy sequences, and all these selected aptamers will be for our further characterization analysis. To get more information of these selected aptamers and for further study the interaction of the selected aptamers and its specific target, we also did sequence structure and prediction analysis by M-fold. As we can see, all these aptamers, the prediction analysis showed that the core region of the selected aptamers showed here the structure from Apta-36, Aptamono 11, and Apta-D4. They showed with they tend to form a stem loop structure here, and we can make a hypothesis that all these this type of stem loop structures they may play an important role for target recognition and for binding capacity, and also for the interactions of the aptamers and for its specific target, different type of methylizing. The measurement is in progress, and we also get good results for specificity of each type of methylizing and these coated aptamers. But here we didn't show the results here. Okay, so finally, I will make a short conclusion and perspective for our further research. By several rounds positive selection and a round negative, we get selected aptamers against the mono, D, and trimethylizing separately. And for our further study, we will study the interaction between selected aptamers and this specific target methylizing. As for this part of job, we are using ITC approach and the mass spectrometry approach and FITC also. And then we will use aptamer-based tool to enrich messenger proteins from wild-type Salmonella and its mutants. And eventually, this work should contribute for us to investigate the bacterium methylome during planktonic and cellular transition from Salmonella typhimurium flagella. Okay, so finally, I have to thank for all the organizations' contribution and support, our university technology campaign, our my scholarship, China Scholarship Council, and our lab, cell enzyme and cell engineering lab, and our organization, CNRIS, and my supervisors, Serafin Padiolo and Dr. Yannick Rossi, and all members of our group. Thank you. Thank you for your attention.